Hello guys, in this video we're going to go through the next topic in the 6.1 notes. We're going to look at topic 2, which is all about the three-dimensional structure of DNA. So a lot of this should be review, hopefully, from the very beginning of the school year. We talked a lot about DNA and nucleotides and nucleic acids. So we're going to recap a lot of things that you guys should already be familiar with. But basically, at this point, in the last video, I talked about these experiments that really helped to prove that DNA is the genetic material. And so in the 1950s, like I said, there was a huge push in the scientific community to learn about DNA. And at that point in the 1950s, they actually did not know the three-dimensional structure of DNA. They did know that DNA was made of nucleotides, that it was a nucleic acid, that was a polymer of nucleotides, and that nucleotides have a nitrogenous base, a pentose sugar, and a phosphate group. But that's basically all they really knew about DNA at that point. They didn't know the three-dimensional structure, like the twisted ladder double helix, like that hasn't been figured out yet in the early 1950s. But by the later half of the 1950s, all of this will be figured out very quickly. And so I'll talk about that right now. But let's start with nucleotides. So they didn't know that DNA was made of nucleotides. Let's review nucleotides real quick. Nucleotides, this is the building block of nucleic acids of DNA. These are the things that get bonded to each other over and over and over again to build a DNA strand. But each nucleotide is composed of a pentose sugar, which means a 5-carbon sugar, a phosphate group, and a nitrogenous base. In DNA, the sugar is deoxyribose. So deoxyribose is the type of sugar found in nucleotides that build DNA. And on that pentose sugar, just to remind you guys, we number these carbons. There's the 1', 2', 3', 4', and 5' carbon. And so you can see that the phosphate group here is attached to the 5' carbon, and the nitrogenous base is attached to the 1' carbon. And then those nitrogenous bases that are found in nucleotides, they could be one of 4 different types in DNA. DNA is either going to contain, or the nucleotides that are found in DNA are either going to contain a nitrogenous base that's cytosine, thymine, adenine, or guanine. They fall into 2 different categories. Some of those nitrogenous bases have a single ring structure, and some of them have a double ring structure. And then there's also uracil, by the way, which if we were talking about RNA, which we're not focusing too much yet on, but uracil is a nitrogenous base found in RNA nucleotides. But in DNA, there's those 4, C and T and A and G. And like I said, some of them have a 1-ring structure, some of them have a 2-ring structure. The nitrogenous bases with a 1-ring structure are called pyrimidines, and the ones with a 2-ring structure are called purines. And then if you guys remember at the beginning of the school year, I gave you guys this mnemonic to kind of help remember which ones are which. So cut, pi, and pure as gold to remember which nitrogenous bases are which. And then at this point, just know or remember that thymine is found in DNA nucleotides, but not in RNA. And uracil is found in RNA, but not DNA. But those are these things right here, like these nitrogenous bases, right? So this nitrogenous base in this picture, I can tell is a purine because it has a 2-ring structure. And so then those nucleotides, obviously they bond together over and over and over again to build polynucleotides or DNA strands. And that involves dehydration synthesis reactions, which join monomers together. And so specifically, we're making a bond. Those nucleotides are bonded between the phosphate group of 1 nucleotide, which is then going to bond to the 3' carbon on the pentose sugar of the other nucleotide. So you form this new covalent bond right here between the 3' carbon on this sugar of one of these nucleotides and the phosphate group on this other nucleotide. And we call this a phosphodiester bond. Or we just refer to this region between the 3' carbon of this sugar and the 5' carbon of this next sugar as a phosphodiester linkage, which is joining these nucleotides together in a DNA strand. And on these DNA strands, you're going to have 2 different ends. And so in a polynucleotide, when you join these nucleotides together, we say that DNA has a DNA strand or a polynucleotide has directionality, meaning that the 2 ends are chemically different. And so on one end of those nucleotides that are bonded together, on one end of those strands, you're going to have the 3' carbon of the sugar But on the other end, you're going to have the 5' carbon of the sugar. So we call this the 5' end and the 3' end. The 5' end contains this phosphate group on this nucleotide, and the 3' end has this hydroxyl group attached to the sugar. But anyway, there's a 5' and 3' end, so make sure you guys remember that. And new nucleotides, when DNA is being made, you guys remember we talked about this before, but new nucleotides can only be added to one of these ends, and they have to be added to the 3' end. So as we start building more and more nucleotides and joining them together, they're going to be added to the 3' end, and that's going to be a lot more important in this unit. So keep that in mind. So now let's talk about the 3D structure. So again, they knew it was made of nucleotides, but then in the 1950s, they're going to learn a lot about the three-dimensional structure of DNA. And so we'll go over a couple groups of scientists here. So there's Rosalind Franklin and Maurice Wilkins. They're going to be investigating a lot the three-dimensional structure of DNA. They're going to use something called X-ray crystallography, which basically just shoots X-rays at DNA molecules, and those X-rays diffract in a certain pattern, and then they can analyze and do a lot of fancy thinking that I have no idea how it's done, but they can look at those diffraction patterns, and figure out some clues about the structure of a DNA molecule. And so they actually found out using these diffraction, these X-ray crystallography images that they were getting, Franklin was actually able to figure out that there was DNA likely consisted of two sugar phosphate backbones, that there was two sugar phosphate backbones in a DNA molecule, and that there was nitrogenous bases pairing in the middle of the molecule, and that the molecule is forming some sort of helical structure. And so that's a huge step forward, obviously, into figuring out the three-dimensional structure of DNA. And then there were these guys named James Watson and Francis Crick, who were also at the same time figuring a lot about the three-dimensional structure of DNA. They actually use, or steal, technically, Franklin's X-ray crystallography images. Very scandalous, but they actually used some of that information from her crystallography to help figure out a lot of the things that they're going to publish ultimately about the structure of DNA. But these guys ultimately are going to figure out the whole shebang about the structure of DNA. They're going to figure out that it's a double helix made of two strands twisted around each other. There's nitrogenous base pairing in the middle. They figure out the actual specific dimensions of a DNA molecule, as you guys can see here. So they're going to really nail down the whole precise structure of DNA. They figured out, again, using the X-ray crystallography from Rosalind Franklin, they helped. They determined that those two strands in the DNA molecule, that they had a uniform width. So like they were very parallel, those two strands in the DNA molecule. And so at first they thought, because they knew there was nitrogenous base pairing happening in the middle between those two strands in a DNA molecule. And at first they thought it would be like purines with purines, or pyrimidines with pyrimidines, or they thought like adenine would pair with adenines and thymines would always pair with thymines. But after figuring out that the width of the two strands is uniform, meaning very consistent, they figured out it couldn't be like two purines together, because purines are the larger double ring nitrogenous bases, and if you put two of them together, that's going to cause the width to be pretty wide. But if it was two pyrimidines together, then that width would be really too short, and so you'd end up with like a very curvy ladder for your DNA molecule. It wouldn't have a uniform width. But if it was a purine always with a pyrimidine, you would have a uniform width that matched the dimensions that they figured out for DNA. So they figured out that actually it's pyrimidines pairing with purines in the middle of a DNA molecule. And specifically, they figured out that it's going to be adenine nitrogenous bases always pairing with thymine nitrogenous bases, and guanine nitrogenous bases always pairing with cytosine nitrogenous bases. So they helped to figure that out too, which they used some of the information that was published by another scientist named Erwin Chargaff. And so there's something called Chargaff's rule that he figured out. This guy figured out. He analyzed the base composition. So they knew they had these different nitrogenous bases in DNA, adenines, thymines, guanines, cytosines, and he actually looked at the percent of adenines, thymines, guanines, and cytosines in the DNA that was taken from different organisms, different species. And so here's some of his data here. You can see that like Here's the percent of each of those bases in like humans' DNA versus like a fruit fly's DNA versus like corn's DNA. And what he found is that the percent of adenine and the percent of thymine were always very, very similar. And the percent of guanine and cytosine were always very similar as well. And that was true for every species that he was looking at. For every species, the percent of adenine and thymine were always very, very similar. And the percent of guanine and cytosine were always very, very similar. And he also figured out, depending on the species, that those percentages are different. And so different species have different percents of these nitrogenous bases, which was also interesting to figure that out at the time. And so anyway, Watson and Crick used that information to help them also figure out that, yeah, it's probably because adenine's pairing with thymine always, and guanine's always pairing with cytosine, which causes these base percentages to always be very similar. Because if there's ever an adenine, well, there's going to be a thymine and DNA connected to it. Or if there's ever a cytosine, there's always going to be a guanine connected to it. So it'd keep those percentages the same. So that was very consistent with the data that was collected here. But that's called Chargaff's rule, which is that the percent of A and T in a DNA molecule is always going to be the same, and the percent of G and C is always going to be the same. And so just to kind of review the 3D structure of DNA, which again, all this was figured out in the 50s, a lot by Watson and Crick, using some of the information collected by other scientists as well. But the DNA is double-stranded. There's two different strands. So there's a strand of nucleotides and a second strand of nucleotides. And each of those strands has what's called a sugar phosphate backbone, with the sugar and phosphate groups alternating on each of those strands. And the nitrogenous bases are pointing in the middle. And those nitrogenous bases there's hydrogen bonds that form between them that's actually holding the two strands of DNA together. And so those nitrogenous bases, when they're pairing together in this specific rule, we call that complementary base pairing. And so G again always goes with C, and A always goes with T. And then specifically, if you look at the hydrogen bonds here, there's a small difference here. Between guanine and cytosine, they form 3 hydrogen bonds with one another. And then adenine and thymine, they are able to form 2 hydrogen bonds with one another. So you can see that here. All of that holding these 2 strands together. And then what else? The 2 strands of DNA are anti-parallel, meaning each of those DNA strands, each of those strands of nucleotides, there's a 3' in and a 5' in, like I mentioned earlier. But if you look at both strands of DNA, they're going to be running in different directions. Those 2 ends are going to be opposite. So in one of the strands, there's going to be one of the strands is going to be orientated in a 5' to 3' direction, whereas the other one's going to be 3' to 5'. So they're going to be running in opposite directions. So for that reason, we say that DNA, the 2 strands are parallel, but we said that they're anti-parallel because those 2 ends are running in different directions, the 3' and the 5' end, which is based off of those carbons in the sugar and which way they're facing. And then lastly here, the last thing in topic 2, I just want to mention some important differences between prokaryotes and eukaryotes in terms of their DNA and the DNA that we find in bacteria cells versus eukaryotic cells, which we'll talk a lot more about the genetics of bacteria and eukaryotes and the difference between them in this unit. But for now, just a couple important differences to point out is that in prokaryotic cells, like bacteria, they almost always have a singular chromosome, just one chromosome. So one long piece of DNA that contains all of their genes and the instructions for that organism, for those bacteria cells. And it's circular. That chromosome is actually circular. It's a circular piece of DNA. So it's kind of like a twisted up rubber band inside the cell. If you were to take it out and unwind it and untwist it, it's actually one giant loop of DNA. Whereas in eukaryotes, well, usually eukaryotic cells, so like, again, think plants and animals and fungi and protists, all these cells that are made of eukaryotic cells that have a nucleus, the DNA is found in a nucleus. Well, there's one difference. And that DNA is arranged into long pieces called chromosomes that contain thousands of genes. But usually in eukaryotic cells, there's going to be multiple chromosomes, not just one. There's going to be many chromosomes. Like humans, we have 46 chromosomes. And those chromosomes, so 46 different pieces of DNA in the eukaryotic cell for humans and one of your guys' cells. And each of those chromosomes is again, one long piece of DNA with hundreds and hundreds of genes on it, instructions on it, but it's linear. It actually has 2 ends. It's not looped. So our chromosomes are linear, whereas in prokaryotic cells, the chromosomes are circular. And then let's see, one last thing on here is just because this is going to come up soon in this unit, There's these things called plasmids that certain cells have. Usually we find these in bacteria cells. Sometimes viruses have these. And then there's some examples of eukaryotic cells containing these. But these are small pieces of circular DNA that have some extra genes on them. They're not technically chromosomes. So chromosomes are these giant long pieces of DNA with thousands of genes on them. But then what we'll find sometimes is these tiny little circular pieces of DNA that exist separate from the chromosome that contain some separate genes on it, some extra genes on these guys. This is not drawn to scale, by the way. Plasmids are very, very tiny compared to like an actual chromosome. But it's these small circular loops of DNA that contain some genes on them. And so we'll talk more about those throughout this unit. But I think that's it for this topic. So hopefully a lot of that you kind of remembered already, or you were reminded of and maybe forgotten. But that's it. Thank you guys.